Now let's turn our attention to the topic of sensory transduction in the olfactory epithelium. So here we have a closer look at the olfactory epithelium, and what we discover are the presence of several types of cells that are present in this epithelium. These spindle-shaped cells, these are the mature olfactory receptor neurons. These are the ones that are going to do the job of sensory transduction. These cells sit within an epithelium that is comprised of supporting cells and basal cells, and some of these basal cells will go on to differentiate and divide, and what we discover is that among this basal cell population is a set of neural stem cells, which from time to time will divide and produce new mature olfactory receptor neurons as olfactory receptor neurons are lost to degeneration or to injury and disease. In addition to the supporting cells, there are glandular cells that produce a thick mucus that covers the upper portion of this olfactory epithelium. So the way odorant molecules interact with our sensory cells is by passing into the nasal passageway and diffusing through this mucus to interact with these olfactory cilia. So these are hair-like extensions from this terminal bulb at the end of the dendrite of this olfactory receptor neuron. And embedded in the membrane of this olfactory cilia then are the olfactory receptor proteins that interact with the odorants. The odorants then will bind to specific molecular receptors that are expressed on the membranes of the cilia, and through a sequence of molecular reactions, the binding of an odorant to a receptor leads to the depolarization of the olfactory receptor neuron and the generation of an action potential at the axon hillock region, where the axon emerges from the cell body. And with the generation of an action potential in the axon hillock, that signal could then propagate along the axons of the first cranial nerve through the cribriform plate and into the olfactory bulb. So now let's consider these molecular mechanisms that generate the depolarization in these olfactory receptor cells. And now let's turn our attention to the mechanisms of sensory transduction in the membrane of that cilia that is present at the apical end of the dendrite of the olfactory receptor neuron. So what we find here is a molecule that we've actually encountered a few times before. The odorant receptor molecule is a G protein coupled receptor. That is, it's a receptor that interacts with an odorant molecule, and on the cytoplasmic surface of that receptor bind a set of G proteins. The molecular structure of this receptor makes it part of the 7 transmembrane class of G protein coupled receptors, the same class that we first encountered when we studied metabotropic receptors for neurotransmitters. We encountered this class of proteins again when we looked at the mechanisms of phototransduction in photoreceptors. The opsin protein that interacts with the retinal molecule is an example of one of the 7 transmembrane G protein coupled receptors. So I think it is so fascinating that this same molecular structure has shown up in so many different places in the nervous system. And in each of these places, the mechanisms involve transducing some kind of an event, be it the binding of a neurotransmitter, the binding of an odorant, or a conformational change in an organic molecule when a photon strikes it, to a second messenger system that is activated through a set of G proteins. And in the olfactory receptor neuron, this transduction cascade involves a G protein that we call G-olf, or that subscript identifies this as a particular G protein that's involved in transducing olfactory signals in olfactory sensory neurons. So the odorant interacts with its receptor, the G proteins are activated, and the activation of these G protein then interacts with a downstream target. And the principal downstream target is adenylyl cyclase 3. And when this enzyme is activated, then second messenger in the form of cyclic AMP is produced. So cyclic AMP levels rise when the odorant interacts with the olfactory receptor. The production of cyclic AMP in turn gates the opening of a cation selective channel. This allows for calcium and sodium to enter the cytoplasm of the olfactory receptor neuron. This leads to the depolarization of this neuron. Now there's even more synergy that happens within this olfactory receptor neuron related to these second messenger systems. As calcium begins to rise, then calcium interacts with calmodulin, and this interaction of calcium and calmodulin then gates a chloride channel, and that channel opens, allowing chloride ions to diffuse from inside to outside of the cell. And as that negative charge leaves the inside of the cell, this amplifies the depolarization of the olfactory receptor neuron. So together, the influx of sodium and calcium ions and the efflux of chloride leads to the production of the generator receptor that ultimately can lead to the activation of our voltage-gated sodium and potassium channels at the base of the sensory neuron where the axon is growing out from the cell body. Activation of those voltage-gated channels produces an action potential that can then propagate along the axons of the first cranial nerve into the olfactory bulb. One experience that I think is common to all of us is the idea of adaptation of our sense of smell. We all know that if we are in an environment where we encounter a particularly strong odor, over the course of the next few minutes, our perception of that odor seems to be much less noticeable to us. While this is a complex phenomenon, sensory adaptation has multiple levels to it. One level is at the operation of the sensory neuron in the periphery, but there are also central mechanisms of adaptation. I want to highlight one known mechanism of adaptation that operates here in the olfactory sensory neuron. As calcium enters the cell, it can interact with calmodulin and produce additional downstream effects. One downstream effect of an interaction of calcium and calmodulin is an impact on this cyclic AMP-gated cation channel. The interaction of calcium and calmodulin can actually reduce the sensitivity of this channel to the binding of cyclic AMP, and this has in effect then a negative feedback signal that is integrated at the level of this cyclic AMP-gated channel, reducing further influx of sodium and calcium, thereby reducing the depolarization with the persistent presence of that particular odorant that's driving this cell's response. Another mechanism of adaptation has to do with this exchanger that we find in the membrane of the cilia. So this is an exchanger that brings sodium ions into the cell and calcium out. So as calcium is exchanged into the extracellular fluids, then that has the effect of downregulating the interaction of calcium with calmodulin. This too contributes to the adaptation of this olfactory receptor neuron to the persistent presence of an odorant. Well, next I'd like to talk about the interactions between odorants and receptors, and this will help us understand a bit of the logic for coding that we find both in the olfactory periphery and also within central structures within the ventral medial forebrain.